All right, so we're beginning here on page 337. What do you notice is way up on the top of page 337? I mean, maybe, no, it really doesn't help you when it says figure, RNA, okay, that doesn't help, sorry. What does it say in the red? That's even better. What process is that? Okay, and what did we say yesterday? What's the important part about transcription? What does a scribe do? Reads and writes. Okay, I think, well, I suppose it can go both ways, but I think you would read something before you write it. Okay, it's not that you're wrong, but I think it's something that gets read first, okay, and then you write about it. Okay, so transcription on page 337. The first step in the central dogma involves, did we read this yesterday? We did, because the term dogma seems, I don't like that. So we get down to what, RNA processing? Maybe that's where we would start today. Okay, are we sharing? I mean, that's fine. And repeat that again? Oh, I'm old, my hearing maybe isn't the best, okay? But regardless, moving forward, RNA processing. We're not going to read the whole part because I was just looking at this. We're just going to read the most important part about it. And then I know you're just shuddering with anticipation, all this information up here. Just, I can't wait. I know. Yeah. Okay. RNA processing. When scientists compared the coding regions of the DNA with messenger RNA that ultimately coded for a protein, they found that the messenger RNA code is significantly shorter than the DNA code. Okay? Then upon closer examination, they discovered that the code on the DNA is interrupted periodically by sequences that are not in the final messenger RNA. These sequences are called intervening sequences or introns. Is that one of your vocab terms? It's in black and bold. I don't think it is. And I'm not saying that it should be, okay, because we're not going to throw it in there at this point in the game. But the coding sequences that remain in the final messenger RNA are called exons. And the reason that that's important, DNA strands go on for millions of nucleotides. So that means that the protein you're trying to make isn't going to be millions of nucleotides long as well, because we just read that's saying, okay, the sequences are called intervening sequences or introns. In other words, it doesn't go the entire length of DNA. Okay, I think the best way to work for this in the process of protein synthesis is using the huddle, the sideline in football. Okay? Like it, don't like it, that's how I felt most comfortable explaining this. You might say, I could not care less about football. Well, that's fine, but it helps me explain this to you. Okay, if you turn the page, okay, that figure 14, those are what codes for each specific amino acid. These three letters here are specific amino acids, and when they go wrong, some of these can be quite detrimental, but some of them are not that serious, because as we look at this, we're going to find out that there's a slight mutation in there that typically the red blood cell should look like this. Okay? And it's supposed to be a circle. Okay? I mean, it might not look like a circle, but that's what it's supposed to look like, because you can kind of get the general idea when you look at arteries and veins, even in your skin, what type of shape are they? What do you think, arteries and veins? Yeah, they're tubular, cylindrical. Yeah. So wouldn't it make sense that a circle would go down a cylindrical shape? Yes. But sometimes what happens, if there's a slight mutation, some blood cells can look like this instead. Okay? Has anyone ever heard of sickle cell anemia? Okay? That's what this means, is that these blood cells look like sickles instead of spheres like this. Okay. So, moving on. Why is there nothing written here yet? That's for you to figure out. That's right. That'll be part of your activity also on Thursday. So with that being said, okay, we know that adenine bonds to thymine and then vice versa. Okay, cytosine and guanine. Guanine, right? Adenine, right? No, I did that on purpose. Just to make sure we're not falling asleep out there. Should be guanine, right? Excuse me. Okay, cytosine, adenine, guanine. Is your X-ray vision working for Superman and Superwoman? Is it Superwoman or Supergirl? It is Supergirl. Because if I'm standing in the way, can you see through me? Oh, you're not Super. Oh, okay. One mystery solved then. You're not Supergirl. Oh, okay. All right. So cytosine, adenine, guanine, adenine, and guanine. Okay. Now, of you two, okay, how does football plays, how do they get from here, which is where? The sideline. How does it get from the sideline to the huddle? So who does that? Okay, so it's either one or the other. It's not both of you, because you're not going to get lost going from the sideline to the huddle, because you're not going to go over to the other sideline. You're not going to tell them your play, are you? You've never done that before, have you? So why not do that? Yeah, they don't even know what you're doing. Yeah, you don't want to do that. Okay? So here's Coach Williamson, or who called your football plays? Okay, so Coach Williamson. He's the boss, right? So in other words, this must be the nucleus, right? Because what does the nucleus do? It directs all cellular activities, right? Okay? So then what we can confidently add then, this is just a semicircle saying this is the boundary of the nucleus, because this whole whiteboard is the cell. Okay? So now, inside this nucleus, we've got the directions. Okay? So which one of you is the messenger RNA? I don't care which one of you pick. Okay? So Braden, you are our messenger, because you're taking the message from here, and where are you going after you get the message? The huddle, okay? What happens if you trip or something? You ever done that before? Then you might have a mutation there. You might have forgot. You said left instead of right. Have you ever done that before? Yeah. I just made that up. You have? Yeah, I just said the wrong thing. Did it work out? Yeah, one time. Oh. just once. Okay. All right. So now a clue here. We know this is DNA for multiple reasons. One, it's got how many strands? 2, DNA is double-stranded. Another aspect, what do we see here? Oh, that's the only time we see that. Well, over here, we see the nucleotide what? Thymine. Does RNA have thymine? No, it's got what? So I could say me, but we did that yesterday. It's got uracil instead of thymine, right? Okay, so now Braden, you're going to start decoding here what the boss told you to do, right? Okay, so basically what we see here, okay, Here's braid in here, this green blob right here. You ever call a blob before? You're a blob, okay? We're just going to call you a blob, okay? So here's blob right here, okay? He's this enzyme that's looking for a very specific place to start, okay? Blob, we're going to start you here, okay? Complementary base pairs are still the same except for uracil replaces thymine. Adenine still bonds to thymine, but it's uracil. Cytosine still bonds to guanine and vice versa. So, with that being said, careful, this is a trick. What is this going to bond to in messenger RNA? You sure? See, it's a trick. Yep. Adenine. What about this here? U. Right about, what about here? So blob is getting his job done here so far. Okay. Is there a better term than blob? It just looks like a green blob. I didn't want to say booger. I just thought blob was better. Blob's better? Okay. So blob is doing his job here. Okay. What's this one? It's supposed to be thymine if it was DNA, but it's not DNA. It's messenger RNA. And I'm sorry, we kind of run out of space here. Okay. So sometimes if you catch on to this pattern, you can just look on the other side and notice that if you have a cytosine on the left-hand side, you can put that for messenger RNA. If you have guanine on the left-hand side, you can put that on the right-hand side. You just have to be a little more careful when it's adenine and thymine because it's uracil that replaces the thymine in messenger RNA. So yes. So why is that important? We're going to start coding for the different amino acids that make this protein. Okay, so one of the things that it said in your vocabulary term, you just wrote down what the back of the book says. No one's going to argue with that, but I want you to look at this and say, well, that makes sense. I can't believe the glossary didn't do that. What did it say? RNA or messenger RNA makes up DNA or something to that effect. Someone have their vocab handy. Just read once what we wrote. What did it say, please? Okay. It was the very first one. I think, yes, I think you had wrote it there, young lady.
Which one is it? Messenger RNA?
Yes.
RNA that carries genetic info from DNA and then it goes to direct protein synthesis in the cytoplasm.
Okay, okay, hang on just a moment. Okay, sorry about that break up in time there. But when we look at messenger RNA or even transfer RNA, we said we want them to come in factors of what? Factors of 3. And this is why. Okay? When you look at that table on page 338, and this is something that you're going to have to be, or you're going to be evaluated on here. All right? On this table, when you're looking at this, in the whatever color this is that red or maroon or red? Okay, so this one's red, then you got blue, then yellow, then green. Okay? So when we look at that, our first letter is A. So we find letter A, that's in yellow. Okay? Our second letter is U. Okay? We go across the top here. Okay? And then we find the letter U, which is in the color red, correct? Okay? Then you go down, and then you find the letter G. Now this, I could understand if you're looking at this and saying, this makes absolutely no sense to me right now. Who is that? Okay? For those of you who have caught on to this, what's the abbreviation for this amino acid? You're right. Just try it again. Methionine. What's significant? What does it say about that methionine? It's a start codon. That is what's starting this whole program. That's where Blob knows where to start. Okay? So Blob is looking for AUG. And what a coincidence. Football starts in August. Huh, look at that. That can't be a coincidence, can it? Actually, it is. That's just a coincidence. So AUG is the starter codon that Blob is looking for to start this process. Okay. The more we practice this, the better we'll be. Let's look at our next codon, which is CG. So how do we do that? Okay. First letter. Here we see the letter blue. Then go across the top, find the letter blue, okay? And then go down and along the side here until you reach letter G. Okay? Huh, what do we see? There's the abbreviation. There it is, right there. Proline. Proline, I think it's proline, and there's no way you would know that. Okay? What is our next codon? Okay, is anyone struggling with how to read this table? I think after about the second or third time, it becomes a little more easy to read. Okay, so our next codon is GUC. Okay, same process. G in the green, U in the red, and then C, just keep going down until you reach it. Okay? And what do we see? Valine. Okay? What a coincidence. Right there. Valine. Okay? And then what is our final amino acid? Yes, there's two more. So it is GGU, glycine, there it is, glycine, okay? And then finally the last one, yep, and it is leucine. For those of you who really like biochemistry, okay, or chemistry in general, how did we like last spring for physical science? Okay. For those of you who really, really like that, when we get to take organic chemistry as a senior, then the spring semester we get to the biochemistry portion, you start drawing these amino acids. We don't do this just to get, I'm going to make them memorize all this stuff. Yeah, maybe it kind of is. But the reason we go through this is because you are going to get asked this in possibly technical school. Well, I'm going to be an electrician. Well, you still might have to take a biology class even in vocational technical school. So you might have to know this information then, and especially introductory biology in college. Okay? So now we've got our amino acids lined up. It was AUG, okay, and then so on and so forth. So these letters are coding for the very specific amino acids because what was your definition of transfer RNA? That was accurate, whoever wrote transfer RNA yesterday.
Type of RNA that transports amino acids.
Right, so what that does is inside the cell physically picks up the amino acid and brings that to the ribosome and starts hooking them in the correct order. Okay? Then the last thing I want you to look at, okay? Look at page 347. Okay? I didn't quite get the terms right because it says these circles right here. That's what I'm trying to point you to on page 347. Okay? Notice that even in sickle cell anemia, the genetic code for that is very, very similar to that of normal hemoglobin blood cells. So chances are there's just one letter off somewhere in that segment, that maybe somewhere in here it was supposed to be this C, but for some reason maybe another U popped up in there. That's a mutation. Something got placed in there that does not belong in there. So then what happens is, rather than having these nice spherical blood cells, you get these that look like sickles. Okay? Now, is that going to be life-threatening? Maybe it used to be years and years ago. Okay? Because I think another topic we talked about was cystic fibrosis. Who's heard of that before? Okay, when I was growing up, if someone was diagnosed with cystic fibrosis, a long life for them was probably 23, 24 years of age. But through the process of genetic counseling, different types of treatments, for those of you who have raised your hand, do you mind sharing how old this person might be now? Are they older than 24? Oh, I thought you raised, oh, someone, who was raising their hand? Am I imagining things? Oh, I did. Does anyone know anybody with cystic fibrosis then? Sorry. Okay. Do you mind sharing how old is this? They're 57 now. So just think of that time frame between early 90s to about where we're at now, 2020. Okay, so 30 years. That expectancy through treatments and genetic counseling, how far we've come. Now, what happens with this, okay, what do you think is the function of red blood cells? They carry something very specific in your blood, because that's why they're called red blood cells. Yeah, they carry oxygen, okay? So when we talk about this, who's going to carry more oxygen? The bottom one, okay? And for two reasons. One, this has a greater volume, but you also have to take into account, if this is your capillary, remember we said it's going down a cylindrical tube, okay? This is going to travel down that very, very efficiently, just the way the man upstairs designed us, or however you want to look at it. Well, what might these types of cells start doing? Tumbling and twisting and turning. What's that possibly going to do in a tributary? Does anyone know, in your geographical terms, what's a tributary? What is the Big Sioux River in reference to the Missouri River? Well, it's smaller. Well, someone said it. So, true, but since the Missouri River flows into the Missouri River, that's a tributary, okay? So what happens is, as these start to tumble and twist and turn, sometimes what can happen, what happens if that blood cell does that? It's going to get stuck and start to backflow. Now, what could possibly happen is through different courses of action, some type of muscle movement, I would think, is going to cause that to backflow again, and then maybe it makes a second run at it, and then maybe it makes its way down there. But it can start to plug those smaller capillaries. So when someone becomes anemic, they're just not getting enough oxygen. Because for one, it's stopping the blood flow, and two, it can't carry as much oxygen because the shape is just off. Okay? So what we see happening, which we're going to practice this. How many feel pretty comfortable with taking a strand of DNA, putting the complementary base pairs in this double-stranded structure, and then putting the complementary message in here, or messenger RNA. Because what we had said before, okay, blob is doing his job here. What are you supposed? This protein assembly does not take place in the nucleus. It takes place in this structure that starts with the letter R. You know it, you just don't know you know it. Just start a cellular structure that starts with the letter R. The ribosome. So this is going to leave and it's going to head to the ribosome. So here's your message. These start to pick up the specific amino acids because of the directions here. So we've got the coaches. Here's the play. Here's blob. He leaves here. Tells everyone where to go. So the plot. I suppose you could say it's the plot. So the play is telling you how all the players are going to line up. So the amino acids are different players on the team. Yes, yes. So, in short, hopefully that makes some sense. And my apologies for you. I don't care about football. Maybe so. But for this, to me, I've had the best, this is how I feel most comfortable explaining this to you. Okay? I suppose you could use other examples. You've got the conductor or the band major, not band major, Ms. Charisma, drum major, yeah, not band major, so drum major, okay? So the drum major is right here because she's taking directions from the boss. Who's the boss? Here's Mrs. Olson. Here's our drum major, okay? But we're not going to use that term, blob, because we don't want to bother anybody. But so the drum major starts to tell everyone this is where you're going to start to line up. Is that, for those of you in band, is that kind of what the drum major does? I know it does the conducting. I get that. But does the drum major, is it majorette instead? It used to be called drum majorette when I was in school.
We call her the drum major.
Just call her, okay, just call her the drum major. Fine enough, or fair enough. So with that, so then the drum major has the directions, goes out to your parade lineup or your band competition, say you need to line up here, and then this is where you're going. Usually, a lot of the band movements were called this. You know what FTL means? Follow something. Follow the leader. That's right. Okay? At least that's the way it was when I was in marching band. You had one person, they better know where they're going, or else you're going to have people running into one another. If you got trombones swinging like this and someone with a trumpet, they're all blowing spit on one another. And maybe, maybe not. Okay. So, protein synthesis. Directions, the code, the person taking that code and lining them up where they need to go. That is, in short, what protein synthesis is. You're assembling proteins to what the body's directing that cellular activity. Some of them, they can go wrong. Yes? Yes? Is there any questions for that? Okay, so I see 10/21, which is on Thursday, is when the vocab is due. Okay, we've kind of got a little ahead of ourselves talking about translation. We'll get that process done tomorrow. Questions? All right, that's all I have. We'll catch up to you next time.